Hello everyone, now we are going to start molecular genetics and molecular biology portions. So I'm hoping to complete this 92 pages just by a quick reading. So let's start. Genome organization, genome. The genome is defined as the totality of all the DNA of an organism. Most genomes, all eukaryotes and prokaryotes and some viruses are made up of DNA, but a few viruses have RNA genomes. The eukaryotic genome consists of two distinct parts, nuclear genome and mitochondrial and chloroplast genome. The amount of DNA present in haploid genome of species is called C value. The amount of DNA present in haploid genome of species is called C value. C value is characteristic of each species. The volume of this video might be to a great extent will be low. So just increase the volume if you need to listen to this. The amount of DNA present in haploid genome of species is called C value. C value is characteristic of each species. The value ranges from less than 10 raised to 6 base pair as in the smallest prokaryote mycoplasma to more than 10 raised to 11 base pair for amphibians, and genomes of higher organisms contain a large excess of DNA. Here we could see the DNA content of the haploid genome of a range of phyla. The range of values within a phylum is indicated by the shaded area. The range of values within a phylum. Okay. To a greater extent, it's higher in flowering plants, lower in viruses. Okay. Amphibians to a greater extent. DNA content of the haploid genome is related to the morphological complexity of lower eukaryotes, but varies extensively among the higher eukaryotes. In lower eukaryotic organisms like yeast, amount of DNA increases with increasing complexity of organisms. However, in higher eukaryotes, there is no correlation between increased genome size and complexity. DNA content of the haploid genome is related to the morphological complexity of lower eukaryotes, but varies extensively among the higher eukaryotes. In lower eukaryotic organisms like yeast, amount of DNA increases with increasing complexity of organisms. However, in higher eukaryotes, there is no correlation between increased genome size and complexity. This lack of correlation between genome size and genetic development complexity refers to a C-value paradox. For example, man is more complex than amphibians in terms of genetic development, but some amphibian cells contain 30 times more DNA than human cells. Moreover, the genomes of different species of amphibians can vary 100-fold in their DNA content. Then genome size in some eukaryotes given. Genome complexity. Genome complexity is the total length of different sequences of DNA. Genome complexity can be measured through the renaturation kinetics of denatured DNA. Renaturation of DNA occurs through complementary base pairing. Renaturation of DNA depends on random collation of the complementary strands and follows second order kinetics. A DNA reassociation reaction is described by the quote half. If the large DNA is sheared into second order kinetics and uniform fragments and allows to renature, then the rate of renaturation of denatured DNA is expressed as dC by dt is equal to minus kc square. The k is the second order rate constant. dC by dt is equal to minus kc square. C is the concentration of single stranded DNA at a time t, and second order rate equation for two complementary strands coming together is given by the rate of decrease in C. Starting with the concentration C naught 0 of completely denatured DNA at t is equal to 0, the amount of single stranded DNA remaining at some time t is C by C0 is equal to 1 by 1 plus k C quote. The time for half of the DNA to renature when C by C0 is equal to 0.5 is defined as t is equal to t half. Then 0.5 is equal to 1 by 1 plus kc0 t half, and thus 1 plus kc0 t half is equal to 2, yielding C naught t half is equal to 1 by k. The product of C naught into t half is called the quote half. It is inversely proportional to the rate constant. Since the C naught t half is the product of the concentration of time required to precede half a greater quote half implies a slower reaction. The reassociation of DNA usually is followed in form of a quote curve. A graph of the reaction, a graph of the fraction of single-stranded DNA reannealed as a function of quote t on a semi-logarithmic plot is referred to as quote produced quote. Okay, curve, quote curve. The rate of reassociation can be followed spectrophotometrically by the UV absorbance decreases as duplex DNA is formed. So a Cote curve describes the kinetics of reassociation of a sample of double-stranded DNA that has been fragmented to smaller pieces, denatured into single strands, and then permitted to reanneal. The Cote half of a reaction indicates the total length of different sequences that are present. This is described as kinetic complexity, and the Cote half of the renaturation of the DNA of any genome is proportional to its complexity. The complexity of any DNA can be determined by comparing its Cote half with that of the standard DNA of non-complexity. Repetitive DNA sequences were first identified by studies of rates of DNA reassociation. What relative rates of reassociation are expected for sequences repeated 1,000 times in genome compared to genes that only a single copy? DNA reassociation is a bimolecular reaction, so the reaction rate is proportional to the square of the DNA concentration. DNA reassociation is a bimolecular reaction, so the reaction rate is proportional to the square of the DNA concentration. A sequence repeated 1,000 times in the genome should be therefore reassociated in the rate of 10 raised to 6-fold greater than that of the single copy DNA. Like squaring. DNA reassociation is a bimolecular reaction, so the reaction rate is proportional to the square of the DNA concentration. Then repetitive sequences, we are going to learn about repetitive sequences. Reassociation kinetics identifies two general types of genomic sequences. Non-repetitive DNA consisting of sequences that are unique. There is only one copy of in a haploid genome, and repetitive DNA sequences that are present in more than one copy in a haploid genome. Repetitive sequences show more rapid renaturation rates than the non-repetitive DNA sequences. The proportion of the non-repetitive and repetitive DNA sequences varies widely. Prokaryotes contain only non-repetitive DNA. Similarly, in lower eukaryotes, most of the DNA is non-repetitive. In higher eukaryotes, up to half of the DNA often is occupied by repetitive components. In case of plants and amphibians, repetitive components may account for up to 80% of the genome. In general, the length of the repetitive DNA component tends to increase with overall genome size. Renaturation kinetics further divide the repetitive DNA sequence into two general classes: moderately repetitive and highly repetitive DNA sequences. Highly repetitive DNA consist of very short sequences repeated. Highly repetitive DNA consists of very short sequence repeated many times in tandem in large clusters. Because of its short repeating unit, it is sometimes known as simple sequence DNA. Such simple sequence DNA can often be separated from the bulk of the chromosomal DNA by shear degradation, followed by density gradient ultracentrifugation in cesium chloride. Equilibrium density gradient ultracentrifugation in CSCl constitute most common method for DNA separation. It separates the DNA on the basis of their differences in buoyant density. In buoyant density is just given in this equation. Like that, pause the video for reading. Then distinctive base compositions of simple sequence DNA and differences in buoyant density result in the formation of satellite to the main DNA band. The sequence of this DNA is known as satellite DNA. For example, when mouse DNA is centrifuged in a density gradient, two types of bands may be distinguished: main band and satellite. A main band contains 92% of genome and is centered in a buoyant density of 1.701 grams centimeter minus 3. The smaller peak represents 8% of the genome as a distinct buoyant density. Satellite DNA are species specific. A single genome can contain several different types of satellite DNA, each with a different repeat unit. These units being anything from less than 5 from greater than 200 base pairs. Satellite DNA sequences generally are not transcribed and are located most often in the heterochromatin associated with the centromeric region of chromosomes. Satellites are present in many eukaryotic genomes. They may be either heavier or higher than the main band. One type of human satellite DNA, the alphoid DNA, repeats found in the centromere regions of chromosomes. Human genomic DNA forms a main band, buoyant density given, and 3 satellite bands are also shown. Mouse DNA is separated into a main band, a satellite by centrifugation through a density gradient of CSCl. Then mini satellites and microsatellites. Mini satellites and microsatellites are randomly repeated DNA and are also classed as satellite DNA, but not appearing in satellite bands on density gradients. Mini satellites form clusters up to 2 kb in length with 20 to 50 repeat units, each containing 50 to about 100 base pairs. So telomeric DNA which in human comprises hundreds of copies of the motif 5 prime TTAGGG 3 prime is an example of a mini satellite. Mini satellite clusters are shorter, usually less than 150 base pair, and repeat unit is usually 13 base pair or less. The typical microsatellite consists of a unit repeated 10 to 20 times. Major classes of randomly repeating human DNA. Satellite DNA mainly at centromere, alpha satellite centromeric heterochromatin, beta satellite centromeric heterochromatin of some chromosomes, then mini satellite DNA. And up to 20 kb at or close to telomere, then microsatellite DNA less than 100 kb, BP base pairs dispersed throughout all chromosomes. Which are the satellite DNA, alpha satellite, beta satellite, microsatellite, and mini satellite. Next is about DNA fingerprinting. DNA fingerprinting, also called DNA testing, DNA typing, or DNA profiling, is a technique used to distinguish between individuals of the same species using only samples of their DNA. The chemical structure and everyone's DNA is the same. The only difference between persons is the order of the base pairs. Chemical structure will be similar. So within a species, the only difference between persons is the order of the base pairs. Hence, every person could be defined solely by the sequence of their base pairs. Although two individuals will have vast majority of their sequences in common, DNA profiling exploits highly variable repeat sequences called VNTRs, or variable number tandem repeat, is a general term which includes microsatellite VNTR and mini satellite VNTR, but it is often used loosely to mention the mini satellite class. Within a species, the nucleotide sequence of the repeat units composing VNTR are highly conserved among individuals. However, differences in the number of repeats and thus in the length of the sample sequence tandem arrays containing in the same sample unit are quite common among individuals. These DNA polymorphisms form the basis of DNA fingerprinting. Just never forget the VNTR, variable number tandem repeat. It's a sort of mini satellite. Okay, the high variability in mini satellites makes them especially useful for genomic mapping, because there is a high probability that individuals will vary in their alleles at such a locus. In human chromosomes, many VNTR regions are broadened by restriction into nucleosides on their sites. Alleles of a given VNTR differ from each other in the size of the segment cut by the restriction endonuclease, giving rise to nearly unique RFLP patterns. This is the basis of the technique known as DNA fingerprinting. Parents here shown, their progeny, cleavage would be happening. Progeny is also shown. Alleles may differ in the number of repeats at a VNTR locus, so that cleavage on either side of generates restriction fragments that differ in length. By using a mini satellite with alleles that differ between parents, the pattern of inheritance can be followed. With this technique, forensic scientists have been able to find missing persons, determine whether or not an individual is the parent or a child, and establish beyond reasonable doubt that a person is guilty of a crime for which he or she has been accused. You are given two unlabeled tubes, one containing human DNA and the other containing DNA of a bacterial plasmid. How could you distinguish this DNA by a reassociation of C0T half analysis? The C0T half curve for human DNA would be more complex than that of plasmid DNA. The curve for the plasmid would be similar to that observed in E. coli DNA, since it consists mostly of a single copy DNA similar to E. coli. In human curve, with its multiple slopes reflecting the presence of different classes of DNA that reassociate at different rates. And moderately repetitive sequences. Moderately repetitive sequences of DNA consist of relatively short sequences that are repeatedly, typically 10 to 100,000 in the genome. The sequences are dispersed throughout the genome. A significant fraction of the moderately repetitive DNA consists of mobile DNA elements, transposable elements, short sequences of DNA that have the ability to move to new locations in the genome and to make additional copies of themselves. However, most of these elements move only very rarely. Transposons are considered as selfish DNA, which are defined as sequences that do not contribute to the genotype of an organism but just propagate within genome. And mobile DNA elements. Mobile DNA elements are the moderately repeated DNA sequences interspaced at multiple sites throughout the genome. Genomes generally contain many varieties of transposable elements. So mobile DNA elements encode enzymes that can insert their sequence into new sites in genomic DNA. The process by which these sequences are copied and inserted into a new site in the genome called transposition. Mobile DNA elements are of two types, transposons and retrotransposons. Transposable elements that transpose to new sites directly as DNA are called transposons. Some transposons move by a conservative process that involve the excision of the sequence from its original position followed by its reinsertion elsewhere. What is that? Some transposons move by a conservative process which involve the excision of the sequence from its original position followed by its reinsertion elsewhere. So conservative transposition involves change in the position of transposon in genome without increasing its copy number. Second type of transposition is called replicative transposition. Earlier conservative one, and then next one is the replicative one. Replicative transposition results in an increase in copy number. In this process, the original transposon remain in place while a copy is inserted at the new position. Conservative in the sense that particular part is not undergoing duplication, but replicative, there is replication process which will be taking place. Okay, transposable elements that are first transcribed into RNA copy of element, which then is reverse transcribed into DNA is called retrotransposons. Wait a minute. Conservative process which involves the excision of a sequence from its original position followed by its reinsertion elsewhere. Two types are present: conservative and replicative transposition. Then transposable elements that are first transcribed into RNA copy of an element which then is reverse transcribed into DNA is called retrotransposons. Here you could see a picture which depicts all this information: insertional sequence or transposon, donor DNA, retrotransposition, flanking DNA, donor DNA, DNA polymerase, RNA intermediate, reverse transcriptase, then DNA intermediates, target DNA, transposed mobile elements. So mobile elements are of two types. Transposons move via DNA intermediate and retrotransposons first transcribed into an RNA molecule which is then reverse transcribed into double-stranded DNA. In both cases, the double-stranded DNA intermediate is integrated into the target site DNA to complete movement. DNA transposons move by cut and paste mechanism, whereas retrotransposons move by a copy and paste mechanism, cut and paste and copy and paste. Later, we are going to learn about bacterial mobile elements, bacterial transposon, of course. Most mobile elements in bacteria transpose directly as DNA. In contrast, most mobile elements in eukaryotes are retrotransposons. Both types generally produce short direct repeats at the site of insertion which flank the mobile element. The length of the direct repeat depends on the type of mobile element. Insertion sequences are most commonly called transposons present in bacterial genome. The general structure as IS element is shown. Inserted repeat, usually containing about 50 base pairs, invariably is present in each end of the insertion sequence. Between the inverted repeats is a protein coding region which encodes one or two enzymes required for transposition of the IS element to a new site. An important hallmark of IS element is the presence of short direct repeats containing 5 to 11 base pairs immediately adjacent to both ends of the inserted element. Wait a minute, that is a new point. The important hallmark of IS element is the presence of short direct repeats. We learned about invert repeats on the two ends, but short direct repeats are also there, containing 5 to 11 base pairs immediately adjacent to both ends of the inserted element. The enzyme that catalyzes transposition of an IS element is called transposase. Never forget that. 5 to 11 base pair direct repeat, protein coding region, inverted repeat, then IS element is also there. General structure of a bacterial IS element is flanked by an inverted repeat at each end. The sequence of the inverted repeats are very identical, but they are oriented in opposite directions. The sequence is characteristic of a particular IS element. The short direct repeat flank the IS element. Direct repeats are not transposed with the insertion element, rather they are insertion site sequences that become duplicated with one copy at each end during insertion of a mobile element. The length of this direct repeat is constant for a given IS element. In addition to the IS elements, bacteria contain composite mobile genetic elements that are larger than IS elements and contain one or more protein coding genes in addition to their which are required for transposition. So genetic organization of bacterial transposon, an example of composite mobile element: defective transposase gene, tetracycline resistance gene. Then we are going to learn about eukaryotic transposons. Eukaryotic in the sense our transposons. Okay. So transposable elements make up 10% of higher eukaryotic genomes. Although eukaryotic transposons similar in structure to bacterial IS elements, retrotransposons generally are much more abundant especially in higher eukaryotes. The P element in Drosophila is one of the best example of transposable element in eukaryotes. This element is 2907 2907 base pair long and features a 31 base pair inverted repeat at each end. The element encodes a transposase. P element do not utilize a DNA intermediate during transposition and can insert at many different positions in Drosophila chromosome. ACDS transposon discovered by McClintock and Mays is an example of May's transposon family called controlling element. The May's genome contains several families of controlling elements. The members of each family are divided into two classes: autonomous controlling elements. Autonomous elements are capable of transposition, and non-autonomous controlling elements. Non-autonomous controlling elements are deficient in transposition. So, along with that, the DS element has an internal deletion and so does not synthesize its own transposase. There are approximately 10 different types of DS element in the May's genome, with the deletions ranging in size from 194 base pair to several kilobases. Then retrotransposons. All eukaryotes contain retrotransposons, mobile DNA elements that transpose through an RNA intermediate utilizing a reverse transcriptase. These mobile elements are further divided into two main categories: viral and non-viral retrotransposons. Viral retrotransposons are flanked by 250 to 600 base pair long terminal repeats or LTRs, similar to those in retroviral DNA, and like retroviruses encode reverse transcriptase and integrate. They move in the genome by being transcribed into RNA and then undergoes reverse transcription and integration into the host cell chromosome. Viral transposons are abundant in yeast, TY elements, and in Drosophila. Then non-viral retrotransposons lack LTRs and they have an AT-rich stretch at one end. In mammals, non-viral retrotransposons are the most common type of mobile element. The most abundant non-viral retrotransposons in vertebrates are the lines, long interspaced nuclear elements, and signs, short interspaced nuclear elements. Signs are 100 to 400 base pair sequence are not contain any genes. Signs don't contain any gene. Signs are transcribed by RNA polymerase 3, and most common signs in human frequently contain a site for the restriction enzyme ALUI1 and consequently are called ALU sequence. This 300 base pair sequences are scattered throughout the human genome, constituting about 5% of the total DNA. A related transposable element B1 is found in the mouse genome. Both of these transposable DNA sequences are thought to have evolved from the gene for 7S LSC RNA, around coding RNA involved in movement of polypeptide in the organelles. The most common line element constitute the L1 line family. Some 600,000 copies of L1 family occur in the human genome, accounting for about 15% of total human DNA. So L1 elements usually are flanked by short direct repeats, the hallmark of mobile elements. The consensus sequence contains two long open reading frames or ORF encoding a RNA containing protein and the reverse transcriptase. Genetic organization of typical line and sign. Line doesn't code for any gene, okay? Now forget that. The shorter open reading frame, ORF1, about 1 kb in length, encodes RNA binding protein. The longer ORF2, about 2 kb in length, encodes a bifunctional protein with reverse transcriptase and DNA encoding internuclease activity. So here we could see a picture which depicts genetic organization of typical line and sign. A shorter open reading frame ORF1, about 1 kilo base pairs in length, encodes the RNA binding protein. A longer ORF2, about 4 kb in length, encodes a bacterial protein, bifunctional protein, which reverse transcriptase and DNA internuclease activity. Later, we are going to learn about gene families. Most of protein coding genes are represented only once in a haploid genome and thus are termed solitary genes. Duplicated genes constitute the multi-gene families. Solitary genes, reversely, duplicated genes are also there. Solitary genes are that duplicated genes constitute the multi-gene family. Multi-gene families are groups of genes of identical or similar sequences. These members are either remains clustered or dispersed around the genome. It may be simple or classical multi-gene families. Families in which all the members have identical or nearly identical sequences belong to simple multi-gene family. The tRNA genes are example of simple multigene family. When a tandem array of tRNA genes, each copy is exactly and almost exactly like all the others. Each cell contains multiple copies of rRNA genes that code for ribosome rRNAs. Even E. coli genome contains 7 copies of its rRNA genes. Human cells contain about 200 rRNA gene copies per haploid genome clustered on one chromosome. The multiple copies of conserved rRNA genes on a given chromosome are located in a tandemly repeating manner. The repeated rRNA genes are needed to meet the great cellular demand for their transcripts. Then complex multigene families. The individual members of a complex multigene family, although similar in sequence, are sufficiently different for the gene products to have distinct properties. One of the best examples of this type of multigene family is the mammalian globin gene. The globins are the blood proteins that combine to make hemoglobin. Each each molecule of hemoglobin being made up of 2 alpha types and 2 beta type globins. In humans, the alpha type globins are coded by small multigene family on chromosome 16 and beta type globins by second family of chromosome 11. The alpha subfamily in humans contain 3 genes. Zeta gene represented only in the early embryonic stage. Alpha subfamily in humans contain 3 genes. Zeta gene expressed in the early embryonic stage and 2 copies of the alpha genes expressed during the fetal and adult stages. Zeta only on the embryonic stage and some other portions expressed of other copies of alpha gene expressed during the adult and fetal stage, not at the embryonic stage. Embryonic in the zeta one. Okay, all these 3 types code for alpha chain. Alpha chain is coded by the 16 chromosome located there. So in human beta globin gene cluster is longer than the alpha chain gene cluster and contains 5 genes. Of the 5 genes, 3 are expressed prior to birth. The epsilon gene is expressed only during embryogenesis, somewhat similar to zeta gene. Epsilon is also like that. Alpha is made up of 3, beta is made up of 5. So that beta chain cluster is longer and which contain 5 genes. So of the 5 genes, 3 are expressed prior to birth. The epsilon is expressed only during embryogenesis, while the 2 nearly identical gamma genes are expressed only during fetal development. The remaining genes delta and beta are expressed following birth. There are several duplicated globin DNA sequences in the alpha. Wait a minute, I will be explaining this once again. Beta, epsilon on the embryonic time, while 2 nearly identical gamma genes are expressed only during fetal development. Gamma, 2 kind of gamma on the fetal development time. Then the remaining gene delta and beta are expressed following birth. There are several duplicated globin DNA sequences in the alpha and beta globin gene clusters that are not functional genes. They are example of pseudogenes. They have a close homology to the functional genes but have been disabled by mutations that prevent their expression. 2 non-functional pseudogenes are present in the alpha cluster and 1 non-functional pseudogene present in the beta cluster. Alpha globin gene subfamily is shown. Beta globin gene subfamily is shown. Just remember chromosome 16, chromosome 11, 3, 5 number of genes. Organization of alpha and beta globin gene family. Epsilon, zeta. These are embryonic phases. Then is about homologous genes. Homologous genes are ones that share a common evolutionary ancestor. A pair of homologous genes are sequences similar but do not have identical nucleotide sequence. It occurs between the 2 genes undergo different random changes by mutation. Homologous genes fall into 2 categories, paralogs and orthologs. Homologous genes present in the same organism are said to be paralogs, whereas homologous genes in different organisms that arose through species divergence are said to be orthologous. Orthologous on the different. Para within. Just remember as para something which is within. In Malayalam, para means forget it. So for example, the myoglobin and beta globin genes of human are paralogs. In Malayalam, para means someone who cheats. So just remember, the only person who can, put you in a trouble will be your, to a greater extent, someone who just you respects or you trust someone. So he is from within your circle. So that person is cheating on you. So paralogs means within, someone who is cheating from within this circle of yours. So just remember it as like this, just for fun. Paralogs and orthologs, whereas homologous genes in different organisms that arose through species divergence are said to be orthologs. Orthologs, different species. Same organism, paralogs, different organisms, orthologs. For example, the myoglobin and beta globin genes of humans are paralogs. Myoglobin and beta globin genes of humans are paralogs. They originate by duplication of an ancestral gene, whereas the myoglobin genes of human and chimpanzee are orthologs. Pseudogenes. Pseudogenes are functionless gene variants that are present as a result of ancient historical accident. Pseudogenes are functionless gene variants that are present as a result of ancient historical accident. Pseudogenes are a type of evolutionary relic that indicates about the changing nature of genome. There are two main kinds of pseudogenes. Conventional pseudogene, a gene that has become inactive, non-functional because of the accumulation of mutations. The globin pseudogenes are most common example of conventional pseudogenes. The human globin gene clusters have 5 pseudogenes. Then processed pseudogenes are also there. A gene that results from integration into the genome of a gene that results from integration into the genome of a reverse transcribed copy of an mRNA. Processed pseudogene might integrate into same chromosome as its functional parent. A gene that results from integration into the genome of a reverse transcribed copy of an mRNA. Processed pseudogene might integrate into the same chromosome as its functional parent or possibly into a different chromosome. Because a processed pseudogene is a copy of an mRNA molecule, it does not contain any introns that were present in its parent gene. It also lacks promoter sequence present in the upstream of parent gene. The absence of these sequences inhibit the transcription initiation and make processed pseudogenes inactive. Then gene. A primary function of the genome is to specify RNA molecules. Each region of the DNA helix that produces a functional RNA molecule constitutes a gene. Although most genes are transcribed into mRNAs which encode proteins, some DNA sequences are transcribed into RNAs that do not encode proteins, tRNA and rRNA. A gene also contains all the DNA sequences required for synthesis of a particular RNA transcript. Thus, in molecular terms, a gene also includes many regulatory RNA sequences which are responsible for ensuring that the DNA is transcribed at the proper time and the appropriate cell type. Total number of genes is known for several organisms. Bacteria is the smallest known genome, M. genitalium, has about 470 genes. Mycoplasma genitalium, great amount, C. elegans and Drosophila melanogaster, Saccharomyces cerevisiae, Bacillus subtilis, Haemophilus influenzae, etc. shown. So size of genes in higher eukaryotes varies greatly. Some genes contain more than 2 million nucleotide pairs. However, genes that are more than 100,000 nucleotide pairs in length are common. Most of the portions of genes in higher eukaryotes consist of non-coding DNA that interrupt the relatively short segment of coding DNA. The coding sequences are called exons and the remaining non-coding sequences are called introns. The RNA molecule called the primary RNA transcript synthesized from such a gene is processed to remove the intron sequence during its conversion to an mRNA molecule in the process of RNA splicing. Introns were first discovered in 1977 independently by Philip Sharp and Richard Roberts. Large genes consisting of a long string of alternative exons and introns, with most of the genes consisting of introns, were discovered. Introns range in size from about 80 nucleotides to 10,000 nucleotides or more. Introns are present in most genes of complex eukaryotes, although they are not universal. Many genes like interferons, histone genes, ribonuclease, heat shock protein genes, G protein coupled receptors lack introns. So introns are clearly not required for the gene function in eukaryotic cells. In addition, introns are not found in most genes of simple eukaryotes such as yeasts. Conversely, introns are rare in genes of prokaryotes. Then organelle genome. Organelles like mitochondria and chloroplast contain an independent DNA genome. Genes not residing within the nucleus are generally described as extranuclear. So organelle genomes are usually but not always circular molecules of dsDNA which code for some but not all of the proteins found in the organelle. Most of the proteins in mitochondria and chloroplasts are encoded by nuclear DNA and imported into the organelle from the cytosol after they are synthesized in cytosolic ribosomes. All mitochondria and chloroplasts contain multiple copies of organelle DNA molecule. For example, each human mitochondria contain about 10 identical molecules. The prokaryotic character of an organelle genome suggests that mitochondria and chloroplasts are the releases of free-living bacteria that formed a symbiotic association with the eukaryotic cell. That is the endosymbiotic theory. You might have learned about this earlier. If not, just search about this endosymbiotic theory. Then mitochondrial genome. Each mitochondria contains multiple mitochondrial DNA molecules. Thus, the total amount of empty DNA in a cell depends on the number of mitochondria, the size of the mitochondrial DNA, and the number of mitochondrial DNA molecule per mitochondria. Empty DNA have been found to encode rRNAs, tRNAs, and essential mitochondrial proteins. All proteins encoded by empty DNA are synthesized on mitochondrial ribosomes. Human empty DNA contains this much amount of base pairs. It lacks introns and contains no long non-coding sequences. Unlike human mitochondrial gene, some plant and fungal mitochondrial genes contain introns, which must be removed by RNA splicing. Human mtDNA encodes the 2 rRNAs found in mitochondrial ribosomes, the 22 tRNAs used to translate mitochondrial mRNAs, and 13 different polypeptide genes. Mammalian mtDNA are transcribed into a single transcript from the major coding strand, and individual products are generated by RNA processing. The mitochondrial genome is much larger in Saccharomyces cerevisiae. In the yeast mitochondrial genome, the genes are more widely spaced than in the human mitochondrial genome, and some of the genes have introns. Then chloroplast genome. Chloroplast genomes are usually circular molecules of DNA. ctDNA are relatively large, usually vary in length from 120 to 190 kilobase pairs. Like mitochondria, chloroplasts contain multiple copies of the organelle DNA, typically 20 to 40 in a higher plant. The chloroplast genome codes for all the rRNA and tRNA as well as 50 to 100 proteins. Then genome mapping, a map of the locations of identifiable landmarks on chromosome is known as genetic map. A gene or DNA sequence having a known location on a chromosome and associated with a particular trait of gene is known as a landmark, genetic marker. Genes were the first markers to be used to prepare the first genetic marks of fruit fly. Mapped features that are not genes but DNA sequences were called DNA markers. There are 3 types of DNA sequences that are commonly used as DNA markers: restriction fragment length polymorphism or RFLPs, simple sequence length polymorphism or SSLPs, and single nucleotide polymorphism like SNPs. First, RFLPs or restriction fragment length polymorphisms. RFLPs arise because mutations can create or destroy the sites recognized by specific restriction enzymes, leading to variations between individuals in the length of restriction fragment produced by identical regions of the genome. A single base change within a restriction endonuclease is a readily detectable genetic marker because the mutated site is no longer cleaved by the enzyme in question. 2 chromosomes that differ by such a mutation are then distinguished on the basis of restriction fragment length polymorphism, which arise because a particular cleavage site is present in only one of the 2 DNA molecules. A mutation that gives rise to RFLP thus represents a genetic marker. RFLP have only 2 alleles. The site is present or it is absent. Chromosome 1 DNA has 3 restriction sites. Chromosome 2 DNA has only 2 of the restriction sites. Then cleavage with restriction enzyme and electrophoresis. Fragment C is the size of A plus B combined. Restriction fragment length polymorphisms. And simple sequence length polymorphisms or SSLPs. SSLPs are arrays of repeat sequences that display length variations, different alleles containing different numbers of repeat units. There are 2 types of SSLP: mini satellites, often used loosely to mean the variable number of tandem repeats or VNTRs, in which the repeat unit is up to 25 base pairs in length. So microsatellites, also known as short tandem repeat polymorphism or STRP or simple tandem repeats STRs, whose repeats are shorter, usually dinucleotide or tetranucleotide units. Then single Nucleotide polymorphisms. These are positions in gene where some individuals have some nucleotide and others have a different nucleotide. And these are vast numbers of SNPs in every genome, some of which also give rise to RFNPs, but many of which do not because of the sequence in which they lie is not recognized by any restriction enzyme. In human, since coding DNA accounts for only about 1.5% of the human genome, most SNPs are found in non-coding DNA, such as within introns and intragenic sequences. Then is about physical mapping. A physical map provides information for the linear structure of DNA molecule. Physical distance is measured in base pairs. Physical mapping uses molecular biology techniques to construct a genome map showing the positions of sequence features, including genes. Examples of most common physical mapping techniques are restriction mapping, fluorescence sensitive hybridization, and sequence tagged site mapping. First, restriction mapping determines the position of restriction sites in a DNA molecule by analyzing the sizes of restriction fragments. A diagram of DNA molecule showing the relative positions of a cleavage site of various restriction enzymes is known as a restriction map. Thus, a restriction map represents a linear sequence of sites at which particular restriction enzymes find their targets. Such a map is generated by subjecting the DNA to digestion with two or more restriction enzymes, both individually and in mixtures. By comparing the lengths of the fragments in the various digests, this is determined, for instance, by their electrophoretic mobiles relative to standards of non-molecular masses, a restriction map can be constructed. For example, consider the 4 kb linear DNA molecule that BAM-H1, BAM-HIND3, and their mixtures cut to fragments in the lengths indicated in figure. When DNA molecule is cut with a suitable restriction enzyme, it is cleaved into distinct fragments. These fragments can be separated on the basis of their size by gel electrophoresis. These fragments produce a series of bands. Each band corresponds to a fragment of particular size, decreasing down the gel. This information is sufficient to deduce the positions of the restriction sites and the intact DNA and hence to construct the restriction map. A small DNA molecule was cleaved with several different restriction nucleases and size of each fragment was determined by gel electrophoresis. The following data was obtained. Read this data. Is the original molecule linear or circular? Draw a map of restriction sites showing distance between sites that is consistent with the data presented. What would have to be done to locate the cleavage sites unambiguously with respect to each other? Circular. It's like this. Cleave with HPA2 plus HIND3. A purified DNA is subjected to restriction internuclease digestion for varying times and it generates partially two fully cleaved DNA fragments. The fragments are separated by agarose gel electrophoresis and stained with ethidium bromide. The bands are visualized with the help of UV light source and photographed. Find out the total number of bands in photographs. Figure is shown. After that, really partial digestion is taken place. Then complete digestion is also happening. So next technique is fluorescence sensitive hybridization. Just look at this figure. This is how it creates. Then fluorescence sensitive hybridization. Fluorescence sensitive hybridization involves hybridizing a fluorescent labeled DNA probe to denatured chromosomal DNA of metaphase chromosome. In the past, probes were labeled with radioisotopes, but nowadays fluorescent dyes are used for increased sensitivity and resolution. In fish, the DNA probe is either labeled directly by incorporation of a fluorescent labeled nucleotide precursor or indirectly by incorporation of a nucleotide precursor containing a reported molecule, which after incorporation into the DNA is then bound by a fluorescently labeled affinity molecule. The position at which the probe hybridizes to the chromosomal DNA is visualized by detecting the fluorescent signal emitted by the labeled DNA. Hence, FISH enables the position of a marker on a chromosome or extended DNA molecule to be directly visualized. STS, sequence tagged site mapping. Sequence tagged sites are short tagged tracts of DNA sequence that is operationally unique and has a single occurrence in the genome. Their location and base sequence are known and thereby They can be used as landmarks in genome mapping. Sequence tagged sites that are derived from cDNA are called expressed sequence tags or ETS. EST. STS are detectable by polymerase chain reaction, are used for localizing and orienting the maps and sequence data. A map representing the order and spacing of sequence tagged sites within a stretch of DNA is known as STS mapping. STS mapping is essentially the same as map distance determined by linkage analysis, except that the map distance is based on the frequency at which break occur between two markers. In genetic mapping, organisms contain far more genes than chromosomes, thus not all pairs of alleles can assort independently on one another. Humans, for example, contain 23 pairs of chromosomes and approximately 3,500, 35,000 genes. Thus, each chromosome contains many genes. Genes that are relatively close together on the same chromosome tend to stay together. If less than 50% of the gametes produced during meiosis in a double heterozygote have a recombinant combination of the alleles, the genes are said to be linked. The linear order of genes along the length of a chromosome can be constructed. This ordering of genes along a chromosome is called a genetic map or linkage map. Linkage map is expressed in terms of map unit. One map unit is the genetic distance in which an average of one chromatid out of 100 will have undergone a crossover event. For in distance less than 20 MP units, the mass map distance is approximately equal to the proportion of recombinant chromosomal products in percent, or 1 cm equal the distance yielding 1% recombinant gametes. Comparison of the actual physical distance between known genes determined by molecular analysis with their recombination frequency indicate that in humans 1 centimorgan on average represent a distance about this. Genetic mapping procedures was first developed by Alfred H. Sturtevant while he was still an undergraduate working in Thomas Hunt Morgan's laboratory at Columbia University in 1911. He prepared the first genetic map of X chromosome of Drosophila melanogaster crossing over between genes located on homologous chromosomes, recombinant genes CD and capital C, small d and C, capital D. Capital C, small d, small c, capital D arrangement. To get this arrangement, the generation of new combinations of genes by crossing over between genes located on homologous chromosomes during meiosis or mitosis.